So Mendel didn't know anything about chromosomes. Mendel didn't know anything about meiosis or the meiotic process. He just knew he had these pea plants that had some really interesting patterns. And one of the things that came from Mendel's work is the law of segregation. The law of segregation basically states that, let's say you're heterozygous, and so you have like a capital A and a lowercase a, those are going to be put onto the gametes randomly. So if you're generating sperm, you're generating eggs, one of your gametes is going to have one of those, the other is going to have the other. And something that comes directly from the law of segregation is this notion of independent assortment, which again, Mendel didn't know meiosis, Mendel didn't know about chromosomes lining up or any of that. So let's jump into it and let's talk about what independent assortment is. So here's a picture of meiosis. And in meiosis, obviously, you have this like crossing over thing that happens, and you wind up generating your 4 gametes. So here are some chromosomes. These are chromosomes. These ones are from mom. These ones are from dad, or what have you. They're not really colored in this fashion. I'm just using that to differentiate these. And let's say we have some genes on each of these chromosomes. And this individual has a big A here and a little a here. This person is heterozygous. Remember, A is like the gene, capital A is like the allele, or B is like the gene, capital B or lowercase b are the different alleles. So let's say we are an interphase, right? So these chromosomes are just sort of spread out. Actually, in interphase, it looks more like this, but I didn't think this was a particularly useful diagram. I just want you to remember that most of the time, if this screen were the nucleus, your DNA is unspooled like a whole bunch of yarn that's just sort of strewn about the room. But let's pretend for a moment that we can actually visualize the chromosomes during interphase. They are going to make copies of themselves during S phase, you should recall. And then during prophase, those segments of DNA are going to condense into the chromosomes that we all know and love. And the homologous chromosomes are going to associate with one another. This is also where crossing over can happen. I'm not going to show that here, but crossing over can happen here. Metaphase 1, these chromosomes line up. Anaphase 1, they are separated. They wind up forming 2 individual cells, and then they undergo meiosis 2, and they wind up forming 4 gametes. If any of this is foreign to you, please go back. Check out previous videos. But we have these 4 gametes here, and each of these has different alleles on it. Okay, so this allele is capital A, capital B, capital C, capital D. This is a very dominant genetic individual, and these are 2 recessive, more recessive gametes over here. Okay, but these ones are all very maternal looking, and these ones are all very paternal looking. And that's not really the only thing that can happen. This is how our chromosomes lined up before. But what was to prevent the chromosomes from lining up in a different orientation? Why can't the chromosomes line up like this, where maybe you have maternal and paternal flipped around? And the answer to that question is nothing is preventing them from doing that. And they often will line up like this. This, however, generates very different looking gametes than we had before. So we go through anaphase 1, we're done with meiosis 1, we go through meiosis 2, and we wind up with gametes that are a mix, with gametes that have contributions from the maternal and paternal genetics here. So you have this gamete is, part recessive, part dominant. This gamete is, part dominant, part recessive. It's just going to depend on how those particular homologous pairs lined up. But, this might not seem like a big deal when you only have a few chromosomes, but again, when you have 23 pairs, when you have 46 chromosomes and you start flipping them and changing the orientation, you wind up with an incredible amount of diversity, which is one of the reasons that independent assortment is important. Independent assortment lets genetics mix and match everything up so that you wind up with much more diversity than you would have had without this process happening. So the kind of the whole point of sexual reproduction is to increase diversity of your offspring. And one of the ways in which meiosis can do this is through independent assortment. The other way in which meiosis can do this is through, you'll remember from before, genetic recombination, crossing over. So, I mean, let's go back. That's what independent assortment is. It's the fact that these chromosomes can line up red on the left, red on the right, red on the left, red on the right. It doesn't matter. The chromosomes can line up in any orientation, okay? And so theoretically, you could wind up with a gamete that's 23 pairs of all the chromosomes you got from your dad, and another gamete with 23 pairs, all the chromosomes that you got from your mom. Okay, that is independent assortment.